We've got some our speakers here from the University of Calgary, from the Computing Science Department. And first, if you're reading this, is Gabby Yurka. And Gabby's an MSc student and in computing science and biochemistry. She's interested in bioinformatics, data mining and social network analysis, and she's currently studying breast as well as prostate cancer. Her advisor is Dr. Rita Alge. He's a professor in the Department of Computer Science and the University of Calgary. He's also a founding editor of a number of, or co-editor of a number of publications and or journals, and dealing with social network and network analysis, as well as network analysis in health informatics and bioinformatics. They brought with him Omar and John, and I think they're going to either run interference or run support. I think it's support probably, isn't it? But anyway, please welcome Rita. He's going to be our first to speak. Thank you.
Good evening. Thank you for having us here to speak about our research. We are working on different aspects of data analysis in the Department of Computer Science at the University of Calgary. We do software development, and we work on different aspects of data analysis. And we are here to tell you about our discoveries related to prostate cancer. We work with the School of Medicine on different projects, from health informatics to bioinformatics. We work with Tariq on prostate cancer data analysis. We work on breast cancer. We developed some software for training, so we may collaborate with Mike in order to bring the training to your house, because we developed some software to help you train yourself if you are at home. It keeps track of your body different indicators for different parts of your body, and it warns you that you have to train your neck, your shoulder, your leg, and it shows you how to do the training the better way. And this software won the Microsoft Imaging Cup Award 2 years ago in Russia. We developed some software for referral system, because you know that the waiting time for patients to see some specialist is very long. We try to optimize this and refer you to the best fitting specialist who can see you in the short time. And we try to collect data from different sources, and we try to combine the results of the analysis of different sources of data so that we can get more accurate and more supportive results. In here, we try to analyze the literature, because different researchers from different parts of the world did their research on their patients, and they discovered different genes related to prostate cancer, to different kinds of cancer. So we analyzed the literature in order to find which genes are discovered in each country, by which research groups are collaborating together, which genes are related to which diseases, so that we can highlight the important genes for domain experts who have wet labs to concentrate on those genes are as the biomarkers, the indicators for the specific disease like prostate cancer. So Gabby will present our findings from literature survey, and we are linking these to wet lab analysis with Dr. Tariq in the School of Medicine. And we are running a survey. We will distribute to you copies of the survey so that we can get feedback from you to discover what are the causes of the disease from the analysis of the data that we will collect from you, and to see whether the disease is local or imported, whether you are coming from outside as a migrant and you came with the disease, or you produced the disease here. So we have different things, and Gabby will describe our discoveries from the literature analysis and will describe also the survey and how to complete the survey. And we expect that you will help us collect the data so that we can analyze the outcome from the survey and combine that with the analysis from the wet lab with Dr. Tariq from the School of Medicine, and we can get better results to the benefit of the community. Thank you. So I will invite Gabby to come and talk.
My name is Gabby, and as David said, I'm a master's student under Dr. Rita and Dr. Rockney. And today we'll be presenting to you one of the projects that we're working on, and then I'll be talking about the prostate cancer survey that Dr. Rita introduced. So the research project that we did is called Combining Data Mining and Network Analysis to Find Prostate Cancer Biomarkers. So first I'll talk about some of the background behind that, and then I'll get into the results. So first we'll talk the background, the research results, and then I'll describe the prostate cancer survey and how it's done and everything related to that. So getting into the background, you might already know this, but 1 in 7 men will develop prostate cancer in their lifetime. And worldwide, it's the #2 most diagnosed cancer in males. However, in the developed countries, it's the #1 most diagnosed cancer in males. And some researchers have wondered, why is that? Is it because in developed countries we're more prone to get prostate cancer? Some other researchers say that the reason for that is because in developed countries they use more biomarker tests, such as the PSA test. So that makes it easier to detect prostate cancer. And the PSA test is more applied in the developed countries. But the PSA test is an example of a biomarker. So what is a cancer biomarker? So it's anything that indicates disease. So it could be a protein, it could be a gene mutation, anything that indicates disease. So let's say, for example, you have a gene that's mutated, but that gene codes for something very important, like how many times a cell divides. If that gene is mutated, then the cell might go crazy and divide a lot of times. So if that gene gets damaged by UV or X-rays, and it's not repaired properly, then that might lead to cancer. Again, you might also inherit a mutated copy of a gene from your parents. So it's all about finding these genes that have the potential to cause cancer if they're mutated. There's some genes that are more sensitive than others because it depends on what they code for. So back to the PSA test, so the prostate-specific antigen. So this is a protein. It's a biomarker for prostate cancer, and it's a protein detected in blood. So it's usually used for early detection, for prognosis, and for treatment monitoring, and you might be familiar with it already. However, one problem with it is that it's not specific or sensitive enough. So you might have higher PSA levels in your blood even if you just have a prostate infection rather than prostate cancer. And the Canadian Health Task Force in 2014, they recommended to not use the PSA test anymore because they were saying that it's overdiagnosing patients, and that means that doctors are going in doing biopsies which might lead to infection, so unnecessarily. So there's a hunt for finding more of these cancer biomarkers, especially for prostate cancer, because PSA test is the best one that we know of right now. So they want to find more really good ones like this. However, finding cancer biomarkers is not easy because there's 25,000 to 30,000 protein coding genes in humans. So as a wet lab researcher, it takes a lot of time to go through each gene and do the right tests to see whether it actually causes cancer or how it relates to the cancer. So how can we narrow this list down? So one of the best ways to narrow this list down for wet lab researchers is through text mining. So text mining is going through existing literature out there in order to find what trends are already there. However, one problem with that is how many articles there are. So how many research articles are there already in prostate cancer? Well, the graph shows you, since 1975, it's been almost exponentially increasing. So the blue line shows the total research articles on prostate cancer. The one in orange is the articles that mention genes, and it's just been increasing. Right now, it's over 120,000 research articles in the PubMed database. So it's almost impossible for a researcher to go through and manually read these articles. So that's why the best way is text mining, which is an automated way to find patterns in the research articles. So there's various techniques that we can use, such as data mining and network analysis that we're going to talk about in order to find these patterns. So some researchers have already worked on this. So they've already looked at how to use text mining for cancer, and these are some common ways that they use it. So, for example, they'll build gene-gene or gene-disease co-occurrence networks. They'll see, which genes co-occur within an article. That means they might have some sort of relation. Does one gene upregulate another gene, or does it downregulate another gene? So you can build a network out of this to see which genes are related to one another. You can also have integrated databases, so these just make it easier for researchers to find what information there is on a gene that they're interested in. And also some of the work, they rank genes related to disease, so they might say, gene A is the most related to prostate cancer, gene B is second most related, based on existing literature. So our approach, what we did is we retrieved research abstracts on prostate cancer, so about 125,000 of them. Then we found the genes within them in an automated way. And then we did some data mining and network analysis techniques to analyze how these are related to each other. So now I'll go through some of the results that we found. So one of the questions that we had was, what are the top countries in prostate cancer research? So these pie charts show the countries that have most contributed to prostate cancer research in terms of the number of abstracts they've published. So United States is the leader. It published 32% of the prostate cancer articles. Second to that is Japan with 4%, United Kingdom with 4%, Germany, Canada, China, Italy, France, Australia, and then Netherlands. And then over a third were published by other countries. Then we also wanted to see what is the difference between these ones on prostate cancer and ones that mention genes. And the result was almost similar. The United States, again, was the leader. And then some of the countries shifted their position a little bit. Japan, China, Germany, United Kingdom, Italy, Canada, France, South Korea, and then finally Australia. So this tells you kind of which countries are giving the most funding for prostate cancer research. Next, we want to see how are these countries collaborating with one another. So the way we found that out was if there were some authors that, maybe one was affiliated with China, and then maybe there was another author affiliated with the United States. If they were working together to publish a single paper, then we considered that a collaboration. Or maybe there was an author that was affiliated with both China and both United States themselves, then again, it counted as a collaboration. So here we can see how the countries collaborated with one another. United States and China had the strongest collaboration, followed by United States with Canada. So there's actually a lot of collaboration going on between the countries for prostate cancer research, but here we can see the strongest collaborations. Next, we want to look at the genes themselves. So what are the top genes? It's kind of hard to see, but these circles, they have a gene name inside of them, and the size of the circle represents how many papers were published on that gene. So the biggest circle there is called KLK3, and that's the gene that codes for the prostate-specific antigen, the PSA protein. So this is the most studied gene, the one for PSA, and that makes sense because this is the best biomarker test we have for prostate cancer. And then we can see the other genes that were studied the most after this one. So AR, an androgen receptor gene, MSMP, KLK2, and so on. What do the colors mean? So we grouped the genes to the country that published the most amount of papers. So you can see a big amount of blue because the United States have published the most amount of papers. But what's interesting is the little orange circles at the bottom, that belongs to China. And that means that China published more papers than any other country on those genes at the bottom. So why would they have, thank you. So I have a laser pointer here. So these ones right here, they're interesting because it shows that China has a particular interest, the Chinese researchers, on these genes. Like maybe it could be because maybe they have a drug coming out for them. Maybe their research is more directed, so they give funding to more promising research in a certain direction. Maybe it could be people in China are more predisposed to mutations in these genes. So here we can see what countries are focusing on more than other countries. Next, we wanted to look at genes that were frequently mentioned together. So not just which genes are the most popular, but which genes are popular together. So we looked at genes that were popular together by ear, every ear mentioned usually together, or genes that were usually mentioned by countries together. So for example, these two genes, A and B, were always mentioned together by most countries. So we can find here relations between genes. Maybe when these two genes are A and B are up together or down together, it could be that it correlates with prostate cancer. So we want to find these relationships as well, like between the genes as well. So these are some coordinate plots that show groups of genes. So if, for example, these genes, if they intersect with this line here, that means that they're a group together that was mentioned a lot together. Then we also did something called soft clustering by year or by country. So soft clustering is when you have some genes that, maybe they're not so important on their own, but as a group they're important. So you want to cluster these together, but also you have fuzzy areas. So a fuzzy area you might think of as it's warm outside today, so it's not either hot or cold, it's warm, which is a fuzzy area in between hot and cold. So similar to here, maybe there's some genes that belong between clusters. So some genes may be clustered together, but there's some other genes that might be between the clusters in the fuzzy gray area. So we wanted to see if we clustered those genes, where did they fall, the most important ones mentioned together here that we talked about in the previous slide, where did they fall in the clusters? So all of them fell in the fuzzy area, so that means these were kind of important genes that were mentioned a lot with other genes as well. So it kind of indicates that these genes that we found in the previous slide here, they are usually mentioned with other genes a lot, so they're kind of central. They could be central important genes, almost like bridges between other genes. Now we want to look at the top 10 diseases. So here we use the web tool, which finds, you give it some genes, and it finds for you the disease that most likely represents these genes. So the top 10 diseases that came up were prostate cancer, of course, and then breast cancer, and also some other diseases such as infertility and mesothelioma, which is a cancer of the internal lining. So that could tell you maybe these diseases have some common genes in common. And breast cancer and prostate cancer, in fact, do have a lot of genes in common because they both respond to hormone therapy. Next, we want to find communities of genes. So communities of genes, the way we formed these communities, we looked at whether the genes were mentioned together within an abstract, a research abstract. So, all the abstracts that we looked at, if two genes were mentioned together in it, we assumed there's some sort of relation between those genes, that those primary researchers had found. So we built a network out of that. And this is the network was much bigger than this. You know, you would not be able to understand it if you look at it. But we pruned it, so we got the strongest relationships. This is the strongest relationships out of that network. And here you can see all the relationships between the genes and the communities that form. So the communities are by different colors. And what we wanted to look for are the genes that have what's called the highest betweenness. So that means that they're bridges between communities. So genes that you would say have the highest betweenness are the ones that lie somewhere between the communities here. Like for example, maybe this one right here, because it's on the edge of this community and it links to these other genes in this other community. So it's kind of like a bridge. Because that tells us that this gene has some relationships to other genes, and we want to find these relationships between the genes as well. So on the right here, we listed the genes with the highest betweenness, or the genes that served as bridges the most. AR, KLK3, again the PSA protein gene, and some other genes as well here. Again, we wanted to find the diseases that most represented these communities. So for each community, we found which disease, using another web tool, represented that, which disease represented that community. So all the communities had cancer as a common disease, and also 3 of them had prostate cancer. And some of the communities had endometriosis as well, which is surprising because that's a disease of the uterus, it's cancer of the uterus. So that is surprising. And obesity, celiac disease, infertility, and diabetes. So these are some diseases that might be related to prostate cancer, and we can examine further what genes they have in common with prostate cancer, or what lab researchers might want to examine actually. So for this research, we presented a new methodology to explore prostate cancer genetic biomarkers. We combined what I described, data mining and network analysis, which was the communities, to find some interesting trends in prostate cancer research. And we looked at some new relationships, such as which genes were mentioned by year, which genes were mentioned by country, and so on. So now for the future, we want to compare results between the prostate cancer results, and we also did this for breast cancer as well. So we want to see the genes that we got for breast cancer and the genes that we got for prostate cancer, and see how they're related. We want to validate with experimental results, such as expression data. So the experimental results, as Dr. Alhaj mentioned, would come from wet lab researchers, from the School of Medicine. So we would validate with actual experimental data. And another thing we might want to do is compare gene locations. So are the genes actually located close together on DNA? Because maybe that also might mean that they're somehow related. And we'll compare it to our results. And finally, we want to do a prostate cancer survey, which might give us another source of data. So to speak more about the prostate cancer survey, it's another data source that we can use to enhance our results. So here we have the research abstracts that I just mentioned. Here's experimental data that we might get from the School of Medicine. And survey results are highlighted here, which is what I want to talk about. That's another source of data that we want to combine in order to strengthen our results. So if we combine it all together, that only makes the results stronger. So to talk more about the prostate cancer survey, it's a survey regarding your body, lifestyle, and environment. So for example, it might ask you, for your diet, how much vegetable oil does your diet contain? So Or how much olive oil does your diet contain? So we want to see kind of those differences, like do you eat a lot of olive oil, or maybe you drink a lot of green tea. So, you know, if you answer as honestly as possible, that would really help us out, because then we can compare the results. We might give the survey to people that didn't have prostate cancer and just see the differences, and then we can, you know, publish these results, and they can be used by wet lab researchers in order to maybe come up with something very helpful in the future for future patients of prostate cancer. So why is the survey useful? So you can find some correlations. For example, let's say we found 2 cups of green tea correlates with lower prostate cancer incidence, or maybe lactose intolerance correlates with higher prostate cancer incidence. These are some examples that we might find, and it might be interesting. And then, of course, we would have to find, you know, what are the molecular reasons for this. So some of the type of questions on the survey, you know, medication, you know, what kind of medications, statins, are you taking a vitamin D supplement, some body metrics, for example, height, weight, and even hair color. It might sound silly, but some research articles have correlated for one reason or another, for example, red hair with higher incidence or lower incidence of prostate cancer. So these are some interesting questions, but it might lead to interesting results. Smoking, smoking habits, diet, as we talked about. You know, do you eat spicy food? You know, do you eat a lot of honey? That sort of thing. Ancestry. So, you know, where did your mother's ancestors come from? Where did your your father's ancestors come from? Where were you born? And disease as well. So, how did your prostate cancer? How aggressive was it? And that's also important to know, to see like if we can correlate the aggressiveness with something else. Exercise, you know, for your job, are you always on your feet, or do you mostly sit down? Geographical location, so you know, where do you live most of your life? You know, is it kind of in the cold Calgary, or you know, do you usually live, you know, kind of in the sunny areas? Like, do you get more sun? So that might be important. So what are we going to do with these results? So the anonymous data, so this data will be anonymous. You know, we won't ask for your name or ID number or anything. It'll be used in aggregate form, so only all together will we consider the data. Like it won't be singled out for each person. And we may use data mining and social network analysis, as we described before, to find some trends. And then the results may be validated by wet lab researchers, you know, using actual experimental data. So we would really appreciate it if you take the survey. So the survey is online at prostatecancer.alhaj.ca. It's a Google Forms survey, so you can take it online. And if you know anyone else that might be interested to take it, whether they had prostate cancer or if they didn't, that would be very helpful if you could pass it along as well. And also we have a hard copy today. You know, if you just want to take it right now and you know, not have to go online later, we can also do that. So yeah, that's everything that I'm going to present that I wanted to present today. So if you have any questions, yep.
I find it very strange that you have stress doesn't figure anywhere, and I personally believe that most of the people that develop prostate cancer, if it's not inherited to families, etc., that it comes from stress. To me, stress is by far the worst disease you can have. You have to get rid of stress, totally. I know I got prostate cancer from stress. I know that for a fact. My family didn't have it, so I know exactly where it came from, and I do not understand why the medical profession do not go into that issue more than they do. I have asked a number of people, oh, well, we don't really know. I mean, to me, I think that's very, very important. to have that question. And how did you develop this stress? You have an answer for this?
Yeah. Yeah, that is a good point. And I mean, I would think that probably the reason for that might be because stress is hard to quantify. Like, I mean, what kind of, you can ask questions like, what kind of job did you have? Like, maybe if you were like let's say a coal miner, maybe your body had more stress than someone else. So we did ask some questions like such as, what profession did you have? But yeah, it would be good to ask about stress, although it would be hard to kind of, some people might not have as much stress as you, but they might think that they did, and they might answer that they did. So that might be hard to compare, but...
So my suggestion is that when you do a survey like that, you should be able to go back to that person and say, okay, what kind of stress did you have? And how did it develop? And how did it affect the other diseases that you got as a result of stress? It might not necessarily be prostate cancer, can be something else. But stress to me is the #1 cause why there are more guys in the US and Canada that prostate cancer than the rest part of the world. You showed it right there, it's #1, right? I guarantee you it's stress more than anything.
Yeah, and actually, once we get the survey results, then we'll go through them and actually might do another survey based on that in order, maybe we want to zero in, like you said, maybe on stress or something like that. We'll ask maybe more questions.
Why wait? Why wait? Why don't you do it now?
No, we will revise the survey based on the feedback from the patients, from the experts. So we have revised the survey several times so far. As we get more feedback, because we are not domain experts, we are not patients, so we don't know exactly what is there. We try to collect information
I'm not your patient
from...
either,
Yeah,
that's okay.
yeah, this is valuable information. Yeah.
But I just think that in doing a survey, why not include it? And because I think that's #1 cause, personally. I know, there's certainly I got prostate cancer.
In the survey, there is a box at the very end for comments and suggestions. So yeah, you can definitely add that there. Like, and if more people agree with that, and then, if more people in this room say like stress, 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 then I guess that will be almost like a survey answer. We'll take it as that, yeah.
I have a comment and question. My comment would be, as a UC computer science graduate and more recently a prostate cancer graduate, I'm excited to see computing power being applied to useful topics like this. Back in the day when I was there, it was simulated ping pong and adding up debits and credits, that kind of thing. So it really is exciting to see it. Now, my specific question was about the data mining algorithms. Does it try and make any sense of these research papers? I know myself reading them, it's pretty hard. Do you just find a gene mentioned with a disease and that's it? Or do you try and say that, get any meaning from what's actually written in the article? It must be kind of complicated.
Yeah, it is complicated, and that's very hard, actually. I mean, it's kind of, you have to analyze, how are two things mentioned in a sentence together, and that's more complicated than whether, they're just mentioned together. So yeah, that is a future work as well. We want to see like the semantic relationship between two things, and there is some research work already on that, and it's difficult to, to get very good results with that, but yeah, we will apply that in the future to see how are two things related rather than that they're related. So yeah, that's a good point, yeah.
Bruce? Thank you very much for your speech. It's fantastic.
Thank you.
Get
your research was great. Your research is great, and then what you coverage is wise, more than what I expected. That you cover different kind of cancers, and on top of it, you have diabetes. So now my question to you is basically, your research now is focused on prostate cancer or any other focus you have in mind?
Yeah, actually the first place we applied this kind of methodology was to breast cancer, and then we did it for prostate cancer as well. And like we mentioned, we want to actually compare the results together to see, like, are some genes for cancer in general, or is it just specific for prostate cancer? Because we do want to find those genes that are specific for prostate cancer, because they would be more effective biomarkers. We don't want something that's too general, which is what the PSA problem has the problem of. So.
I would like just to add something to that. The problem is we are computer scientists. We are not I'm not a biologist, so we depend on if you find a domain expert, we work with him. First, we found with the Dr. Dimitri breast cancer. We work with that. Then we started working with Dr. Tariq. Now we are focusing a lot on prostate cancer because we have his expertise in doing the wet lab. So currently we are focusing on prostate cancer. We depend on domain experts with their availability and their expertise to go in that domain.
I understand your question, but that's the reason I really appreciate your research and I find the potential phenomenal. And then you cover so many areas. My interest, personal interest, I know you focus on prostate cancer or you some other thing. You know what I mean?
Oh yeah. Well, yeah, I mean.
Depends on the research grant, I understand that. But I'm asking you now is, what is your focus?
Yeah, I guess the focus of the lab is breast cancer and prostate cancer, these two, yeah, for the most part. And some of the other diseases that we mentioned, it was kind of, we found with that web tool that those diseases were popular for some of those groups of genes. But our main focus is, for this research project, prostate cancer. And we have another project that we're working on with Dr. Tariq, that's prostate cancer as well. So.
Any other questions?
So, sorry, the big gene was the one the United States working on, Canada's working on, and he has the other ones down in China's working on.
Yeah.
Well, what would they be working on those ones compared to, I guess it was a PSA one is a big one, right?
Yeah.
And these ones down here that you had on that slide, why would they be working on those ones?
Yeah, so.
More than the other ones?
Yeah, so those genes, I'll go back here. Those genes at the bottom there, that means that China published more papers on those genes rather than United States did, and which is a bit surprising because United States, published the most papers out of everyone. But we don't know yet. But one guess could be that perhaps they have maybe some drugs coming out for those genes, and in some countries they direct the research more than others. Like maybe in Japan and China, they focus the funding more towards certain things. So that could be one reason. Another reason could be perhaps maybe the Chinese population has a higher disposition to have a mutated gene that lies in one of these genes. So that could be another reason. Maybe the Chinese population is more predisposed to mutations in these genes, or perhaps they have some sort of drug that's promising for those genes, or related to those genes. Yeah.
And there are different families of genes that do the same task. So might be in China these genes are similar to those genes in US, but we have to depend on the main expert to analyze this and find out whether these genes are doing the same function. And this is active in China, this is active in the US because of the weather conditions, the eating habits, the environment, every other thing may affect the expression of the genes.
By population, do more people get prostate cancer in China than in North America? Do you know that?
I'm not sure for those statistics. Yeah.
It's actually really interesting that they're trying to piece out dietary influences, and they find that Japanese, Chinese, people from Asia, when they immigrate over here, they also adopt our increased risk for prostate cancer. So they still can't figure out exactly what sort of dietary patterns for sure they are, but there's something about what we do here that puts us at an increased risk.
Got to drink more green tea and eat more rice.
That's why the survey is actually very general, because we have like a kind of a big targets, and we want to kind of narrow in of some of these differences of what gives people a higher risk for prostate cancer. So that's why the survey covers so many different topics, just to kind of narrow in on something.
Any other questions? Thank you.
Thank you for having us.